Why do we think that metabolomics is a great tool to stratify patients and guide therapeutic decisions? For that intro, we will touch upon the traditional drug development process, how a process driven by precision medicine would look in comparison. We will address the need for patient stratification, as well as the impact that metabolism has on therapeutic response. Finally, we will look at a current example of how metabolomics can drive findings and hypotheses for therapeutic modulation, in this case with histidine. If we look at traditional drug development, I think we all know that it's a pretty streamlined process based on established knowledge, but the failure rate is pretty high of up to 96%. In a published example from AstraZeneca, it has been shown that from 2004 to 2010, their success rate from drug candidate to approval was just around 4%, in spite of annual investment of £5 billion. Most targets fail in the clinic either because there are safety concerns or because the drugs are not efficacious. Here, metabolomics could be insightful in various ways. It goes from mode of action studies in cell lines to patient stratification in clinical research. Now, AstraZeneca is a model in how precision medicine and omics could drive success in clinical stages of development. They acknowledged that the main reason why drugs fail today is that we just don't understand the underlying pathophysiology well enough. So AstraZeneca has started to invest heavily in human-centric trials and hypothesis-free discovery. So what they do is do multi-omic studies that allow them to fine map omics data to the clinical phenotypes that they observed, and that only builds the basis for all of the other steps in the development process. And in the clinical stages, they always use a personalized healthcare-driven approach. These changes in the process have led to an increase in the success rates from 4 to as much as 30%. So really an astonishing result. Now, let's briefly talk about, is better patient stratification needed? And I would assume most of the people here in the webinar would subscribe that it is necessary to do better treatment stratification. Let's look at 3 areas where this is really needed. First of all, non-response is an issue that affects virtually all types of pharmacological therapy to a greater or a lesser extent, whether that's metformin in diabetes, where the response rate might be around 2/3, or Copaxone in multiple sclerosis, where the number needed to treat is much, much higher than that. We believe that metabolomics can be used to stratify patients based on their biochemical phenotype, and that this is much more informative than what we have in routine biochemistry. The second area where metabolomics can be helpful is therapy monitoring. And again, no matter whether you're looking at diabetes, depression, inflammatory bowel diseases, or cancer, drugs do tend to become ineffective, whether this is through resistance mechanisms in cancer or just disease progression in diabetes. In those situations, genetics is often not very useful because by and large, the genome is stable. On the other hand, metabolomics can cover the dynamics of a disease across the patient's journey, so you can more easily monitor whether the drug that you're giving is still the right drug to use. The third area is lack of actionable biomarkers. I have been to a conference on CDX lately, and the message there was very clear. Having a CDX drives success rates in the clinics. For cancer, it's about 2 times as much if you have a CDX in place as compared to if you don't. But all the other indications are lagging behind, and there's not so many actionable biomarkers in those indications. And it's not just about selecting the right medication, it's also selecting the right co-medication, nutrition, lifestyle, and probiotic therapy recommendations that are now pretty much based on trial and error. Here we believe that knowing the biochemistry of the patients and knowing the biochemical mode of action of the interventions that you might want to use really drives precision medicine. Coming to the last part of my introduction, how does metabolism impact therapeutic response? In a typical patient cohort, you will have something like depicted here in the middle. You have some responders, but you also have a substantial number of non-responders. And if you look closer, you have subsegments of patients that are primarily non-responders, and you have other subgroups of patients that respond much more favorably to a drug. And the reason is that there's some sort of suppressor in one group, like antibiotics, and there's some sort of booster, like immune modulating therapy, in the other patient groups. For the response suppressors, I think the most well-known example is antibiotic therapy in immune checkpoint inhibition, where it's been shown that patients who have not been on antibiotic therapy had a much greater benefit of immune checkpoint inhibitor therapy as compared to patients who had used antibiotics briefly before. The plausible mechanism is very much metabolism based, because it's well known that antibiotic therapy alters the bile acid pool as well as other microbiota associated metabolites that are known to be immune modulatory. It's not just antibiotic therapy either. About a quarter of the most common drugs have some sort of antibiotic function, and that includes anti-inflammatory, antihypertensive, anti-diabetic therapies, or proton pump inhibitors that many of the patients that are treated for whichever complex disease will take at any given time. An example for a response booster is shown here, and this is a study based on biocrates technology, where researchers from Heidelberg have found that patients that have higher levels of certain types of lipids with long-chain fatty acids were much more likely to respond to immune checkpoint therapy in urological cancers than patients with low levels of those very long-chain fatty acids. It is believed on a mechanistic level that those very long-chain fatty acids promote peroxisomal function and thus anti-tumor immunity. The idea that was generated based on this study was that patients could be supplemented with those very long-chain fatty acids and thus boost the response. And it would actually be pretty simple, because those lipids are contained in fatty fish or in fish oil. On a more general level, we think that metabolomics can serve as performance companion diagnostics tool by informing you whether a specific drug is going to work in a patient, by helping you decide which is the best drug for a patient if there's multiple treatment options available, and also which co-medication or other types of supportive therapy you could give your patients. So in this case, you would look whether a patient is a predicted responder, then you would start therapy. And if the patient is a predicted non-responder, you would either decide to use a different therapy or use a supportive therapy that is designed to boost the response. And as we've mentioned before, it's easy to reanalyze the metabolome. So it's a perfect tool for the disease monitoring or treatment monitoring as well. Finally, here's a very recent example published just a week or so ago, where a group has done plasma metabolomics in both humans and a mouse model of pancreatic cancer. What they have found is that there is vast changes in the metabolome in pancreatic cancer patients, and that included low levels of histidine. So here the researchers had the idea to supplement histidine, and they have done so in mice. And as they had expected, supplementing histidine together with chemotherapy has prolonged survival and also led to a reduced tumor volume. You see that depicted here. The green line is the mice treated with high level histidine nutrition and gemcitabine. It has also been confirmed on cellular level that histidine supplementation increased cellular histidine levels and also altered other metabolites. So we can conclude here that metabolomics provides insight into disease pathophysiology and also generates hypotheses for pharmacological, lifestyle, and nutritional therapy approaches. With this, I would like to close. If you have any questions about my presentation, please post them and we will answer them later on.